thanks. So we move on to the next talk. The common talk, or at least we have one speaker for LSCE and another one for BIAM. So we will start. It's about acclimation for vegetation climate change. So we will start with Jonathan Barichivich, which is with us from Chile. Hi, Jonathan. So I don't know how do we do Nala? Is Jonathan sharing his slides?
Azila, we can share here and move to the other slides. Jonathan,
Oui.
do you have a question or?
So can you slide or
yes. Yes.
You will share?
Okay. Yeah, you can share, and I think I can move, or I say next probably.
Okay, so we share.
Yeah.
So we start the share. Trying to start the sharing. And you have 10 minutes.
Okay.
It's up to you.
Okay. Hello, everyone, and I'm in the field now, so I'm from far away, but I will try to show you a bit what is a bit the work that my team is doing in regarding acclimation. Is not the main target, but are sort of aspect of our research that involve acclimation, and I hope we can find very interesting complementarities to work on this part. So I'm a director of research au CNRS, section 30, and my research program is to advance process-based understanding and modeling of forest ecology in the earth system, but at long time scales. So I'm interested in adding new type of observations or constraints to the model, to ORCIDE, but at long time scales, from decades to centuries. And I was awarded recently an ERC starting grant, which is called CATES, which it's about the consequences of altered tree growth and physiology in the earth system. So, and I tap a lot on tree rings in this project. And next. And so just to start a bit, to set the stage, so there are some concepts there that are interesting to define. So when we think about adjustment of ecosystem or plants, individual plants to a changing environment, the first thing is that what we call phenotypical plasticity, or simply plasticity, which is just the ability of an organism or an ecosystem to change traits in response to environmental changes over a lifetime. So that's very generic concept, but then within that there is what we call acclimation, which should be these short-term reversible adjustments, so temporary adjustment in traits, in some traits, in response to a change in environmental conditions. For instance, what we usually do is the acclimation to warming, acclimation to drought, and this means that there is an increase of tolerance to a stress level from an environmental factor. For instance, when we look at acclimation of photosynthesis to warming, the optimal temperature for the Farquhar model, for instance, we have to account for those adjustments that plants tend to do at short time scales. And then there is the other concept, which is adaptation. Sometimes one can mix both, but adaptation refers to long-term, at the evolutionary scale, changes in a population to adjust to environmental changes. That would be basically through genes, changes in different generations of an organism. So it's not just over one generation. So that's the difference. And this type of adjustment of short-term or long-term interact and also are important because impact on how ecosystem really track the pace in climate change and is central for modeling the processes in the air system, which basically with land surface models that predict, or we use these tools to predict the future state of the air system. Next. So in this, it's very brief what I will show, but it's the points where we can probably collaborate, and it's not an exhaustive survey over what we are doing that touches acclimation over the whole LSE. It's just a very small view of more the immediate team, but there is much more to what I'm showing. Yeah, that one. That's a good one. Yeah. So when we think about the ecosystem acclimation here, there is an axis on the impact of processes. So acclimation is mediated through changes in some processes in this case. So, so the impact, there is one axis which is the impact on ecosystem function of that process, and then the time scale in the horizontal axis. So there are processes there that we can think that are really fast, short term, which is like, for instance, the acclimation to GPP, acclimation of phenology or LAI. And then we go to more intermediate time scale, like changes in demographic, which go from probably years to decades and centuries. And then changes in stand density as well, that can, for instance, with increasing CO2. But as we go to longer time scale, it becomes very difficult to measure, very difficult also to constrain and to model. We really don't know what happened at those time scales. And those time scales have a big impact on the overall ecosystem function. That's the idea. So biomass allocation, and then nutrient availability, for instance. So it would be the nitrogen cycle or the phosphorus cycle at long time scales. And what happened in the end is that this usually in these short-term processes, they track climate change, the pace of climate change, but the long process toward the right, they tend to lag behind climate change. And basically that's become very difficult to model. And next. And so then we see that acclimation is poorly represented in land surface models like Orchidea, because parameters are fixed. So they are not time variant parameters. They are time invariant. And that poses a problem, because if we want to predict the future responses, we should really incorporate these adjustments at short time scales, and also not only short time scale, like as we do often, based on physiological acclimation, using or parameterizing the acclimation of GPP and respiration, for instance, to temperature and route, which is being done in Orchidea and in several land surface models. That's the physiological acclimation, basically, is what we incorporate in this model, but the other processes are not, there's no acclimation simulated in those processes. And it's even very difficult to measure acclimation at those time scales. Next. And so for that, here is where true rings come in, basically, because in the panel A left top in blue are how the global change factors change over time. So from 20th century. Then on the B panel is how we go from a cascade effect from climate to really a true ring. So all the processes are involved, which I call physiological dendrochronology, and that we usually tend to measure in some monitoring site. So those are the processes that we measure and then that we model. So to link climate to true ring, and we go through biophysical processes and microclimate, photosynthesis, allocations, and then in the end wood formation, and then the true ring. So what we see with true ring is that on the panel C, to the right in green, we see that over the 20th century, usually width density tend to decrease a bit. And we see that true ring width is very complicated in the stem. Basically, in some places increases or decreases, but overall is sort of flat. But when we go to, for instance, water use efficiency at the bottom, we see that there is a strong increase over the 20th century, particularly as CO2 has increased. But then over recent decade, there is a sort of decrease in the rate of response. So there is a bit of a flattening of the response. And that comes from isotopes in tree rings in wood year to year. So the advantage of tree rings here is that on the right, we see that tree rings are more than just ring width. So there is different dimensions to this. And so there is, for instance, xylogenesis, which is the study of wood formation at weekly time scale at the cell level, the different tissues and the maturation of the cells. And that tells us how biomass is being formed, and also how it is, this process is responding to changes in the environment. And then we have isotopes that tell us about water use efficiency and basically what happened, stable isotope, and what happened at the canopy scale, like water use efficiency, for instance. And also can tell us the source water that plants are using, like with the oxygen stable isotope, delta OET. And then finally, we also have some clues on allocation. So if we look at the root rings with the stem rings, we can get some insights on allocation. And that can go from seasonal to centennial time scales. So here is where we start to really get these long-term metrics. Next. And so using tree rings, we are with Valérie Daw and Philippe Pelin, we are using an approach which is using rural to urban gradients in some mega cities. So we have a rural to urban gradient in Paris that goes from Fontainebleau Forest, 60 kilometers south of Paris, towards the interior of Paris. So to Parc Moncerise here in the picture, where there is a meteo station that is one of the longest in Europe. And then we have oaks. So we have been looking at oaks, and we did tree rings, the classical tree rings, and also isotopes to look at the physiological adaptation and responses to drought and heat wave, for instance, of these trees along this gradient. So we compare the ends of the gradient, and by comparing this, we expect to get some ideas of acclimation to warming, because trees growing in the urban heat island, compared with the rural trees at the beginning of the century, their temperature difference should be smaller than now, and so there is like a warming experiment, and we hope that we can pull something out of that, and this is work in progress. Next. Then we also are interested in acclimation leaf function in very ancient trees. So here is Lañilawal. It's a tree that is in southern Chile, that is about 5,000 years. So the thing is that we use tree rings again to study the function of this tree, but we have been looking at the leaf, and we find that basically even in young leaves, which are about 5 years old or 7 years old for this tree, there is most of the stomata are basically covered by wax. So wax plaques in the stomata, and that means that even it's like the tree is somehow reducing the functionality of the leaf. So even when we have a leaf area increase every year as the tree grows, there is a large part of that basically is rendered not functional. And we want to see that, how it works, and what is the function of this of this process for this tree. Basically, these affect stomatal conductance and directly, and photosynthetic rates. Next. And so here is an example of this tree. And so there's CO2 on the x-axis and ring width in the top, in the vertical axis. And so we see there that basically as CO2 has increased over the 20th century, there was an increase in ring width and biomass growth. up to 1990s, when the maximum temperatures and the vapor pressure deficit start to increase a lot. And basically this is accompanied by drought, more a drying climate, and also drying atmosphere, basically that neutralizes this former positive response of the tree in recent years. And so all these next.
1 minute, Jonathan.
Yeah, I'm finished. I'm finished here.
So we
And,
switch now to Nathalie, and we will ask a question after.
Okay.
So Nathalie, the floor is to you.
I can share my screen.
Okay, go on.
It's working?
Yes.
You have the full screen or?
That's it. Soon.
Here we go. Is it working?
Yes.
Okay, thank you. So first, thanks a lot for the organizer for this day. And I'm sorry to not be here because anyway, so in my presentation, I will be happy to be here. For my presentation, so I would like to present what I'm doing in our research institute on my project, but and what my main focus is, I try to understand and to understand the mechanism and the molecular physiology of the stomatal regulation, and how the gas exchange are controlled in the plants. And for the presentation of today, I try mainly not to go in detail of what I'm doing, but I'll try to establish a link between what I am doing and what could be interesting for the LSCE, or at least what I know about it. And what I heard so far for the modelization, I think it would be very interesting because I'm not familiar at all with modelization. So as you know, probably, like, plants make continuum between the plant and the atmosphere, and from the soil, from the water to, so the water grows through the roots, and then as a water, and then goes through the atmosphere, through the leaves by vessel, and go through the atmosphere, through the stomata. And the water was liquid at the first, and then become a vapor through the stomata. And so what I am interested in how to understand how stomata are controlled and how the gauge exchange occurs through the stomata. And mainly I'm interested to understand what the environmental factor control the stomatal conductance. So we know that probably everybody knows that stomata are open during the day, and they control the gas exchange between the plant and the atmosphere. And during the day, there is the influx of CO2 and it's going to the chloroplast for the photosynthesis. And during the night, the stomata are closed. And so if we look at the stomatal conductance and the CO2 conductance, the first thing you can see is, like, during the day, of course, is you have an increase of the water loss from the plant, but you also have a lose of water during the night, a little bit during the night. And you can see that, of course, you have an influx of CO2 during that time. And what is very interesting is that there is a challenge between the influx of CO2 for the photosynthesis, and which is important for the growth of the plants. And this is a very trade-off between the CO2 influx and the water loss. And each time the plant takes few molecules of CO2, they lost many molecules of CO2. And the rate between these two elements are very important. So what is one of the parameters which is very important for the plants is the stomatal conductance, which is called GS, which represents the exchange between the CO2 uptake and the transpiration through the leaf. So we know that the climate of the Earth will and will be influenced by the vegetation expansion. For example, since the apparition of stomata, like 400 million of years, we know that at the beginning, the structure of the stomata, the first thing is that the structure of the stomata was still very similar with ancestral plants and now. And if you look at the evolution, we can see that the structure will be the same, but the density, the distribution, and the conductance evolve. And also that there is a feedback of the vegetation on the water cycle. So it's very interesting to look at the evolution of this stomata. And we also can see that depending on the environmental conditions, so the water, the precipitation, and the CO2 concentration, that the number of stomata, the density, the shape of the stomata, but also the kinetics of the opening and the closure of the stomata change during the evolution. And this probably influenced the climate. And this is a yin and yang because they are evaluate. There is effect of one of each other. And here, another point, what already mentioned, is like the conductance evolve, and it's very linear if you look with assimilation with the CO2, so with the photosynthesis. And you can see that at the beginning, when there was ancestral species, there is no big effect on the photosynthesis. But then here today, we have, if you look at the land plants now, they are have more stomata than before, but the size are smaller, and they are very important for the photosynthesis. So we know also that the global change, and I will thank the very recent paper coming out from the group of Yang, they show that there are many environmental factors that are involved impact the stomatal conductance. So for example, the increase and the decrease of precipitation, the elevation of CO2, the concentration of ozone, nitrogen deposition, and elevate temperature. And if they are looking at the large level, how they affect the stomatal conductance, it's very interesting. And this is something that I'm working on to try to understand how we can adapt to the climate change, but also how we can make plants more tolerant to this rapid evolution or drought stress, for example, in the case of agricultural plants. So in the paper, that was very interesting because they show that they use like more than 5,000 pair of treatments. They use they extract the data from the literature and look like around more than 450 species. And they saw that the stomatal conductance can be really affected. And this work was done around 30 years before. So they start the study, they took all the data from since last 30 years. And they show that the change in the CO2 composition, the elevation of CO2 in the atmosphere will reduce more than 10% by 100 ppm of CO2, the stomatal conductance. And they did everything for that, for the temperature, precipitation, and ozone. And it was quite interesting because they look and they can look for one event, for example, for one change, but what will be the response on the stomatal conductance? Because we know that the climate change will not involve only one parameters, but probably but affect many factors. So the question is how this response of stomatal conductance will be affected by multiple combination. Do the effect will be additive, synergic, or antagonist? And here now, since several years now, in our lab, we are very interested to understand the physiological and the molecular mechanisms which is involved in the stomatal regulation. So at the first point, we are interested to understand the physiological mechanism, but also to go deeper in the molecular mechanisms. to identify some important gene and to identify what will be very important for the stomatal regulation, we have developed genetic screens, me and others, in several groups, and we were able to identify some mutants which are affected in stomatal regulation. So, for example, we have some, we can have mutant which does not respond to light or does not respond to CO2 or draw stress. And we can isolate some specific genes which are involved in this regulation. So we have now several very interesting mutants that do not respond. For example, we have a very high stomatal conductance, or we have mutants that do not respond to drought stress, but also some mutants which are affected in stomatal density and or stomatal shape, and this one are also affected in the stomatal conductance. So using different, we isolate this mutant, and we have characterized most of these mutants by using several approaches. So in our lab, we are mainly using infrared camera, which is give you a report about the lift temperature and give us an idea about the stomatal conductance. But we can also grow our plants in a very controlled condition, and thanks to the phytotech platform. And we can also measure the gas exchange, the conductance by gas exchange. Of course, in contrast to what I heard so far, we are mainly working with one plant, and the idea is to have the control of the stomatal conductance in environmental condition in only one one plant, and we are mainly using Arabidopsis, which is our model plants. And using this this parameter and these mutants, we can calculate also and modelize the stomatal aperture. And for us, it's a great interest for trying to improve the kinetics of the stomatal aperture, because we know that it's very important, especially for increasing the water use efficiency, but also for the photosynthesis activity. So for coming back to what we have done so far and by identifying key regulator of the stomatal opening or stomatal regulation, now we can try to use this mutant and to look at not only one parameter or the response to one environmental factor, but the idea is to try to found or to understand how stomata are regulated by several environmental factors such as light, CO2, and also a drought stress, which is main involved the phytohormone acid abscisic. So just to summarize, the idea is like, what will be the impact and how stomata can sense and adapt, and how we can improve the kinetics of the stomatal regulation in response to CO2, light, and temperature and humidity, and to improve or to understand the impact of carbon on photosynthesis and transpiration. And just to do that, the idea will be to challenge wild-type plants and mutants which are effective in one of these response to evaluate the impact of this parameter on the stomatal conductance and also on the phenotype, on the growth of the plant and on the photosynthesis, and to get some stomatal parameterization in the global model. And if we can have that on Arabidopsis, then we can improve or use this parameter on other species to try to validate. And since in the Institute we have developed a new chamber, which is called IMAPLAN, where we can follow and we can be able to tightly control the environmental condition with temperature, light, and CO2. We can follow the photosynthesis activity and the conductance. It will be a very nice tool to challenge the plants and to get a new value for stomatal conductance in response to the environmental factor. And the idea will be that I'm convinced that since we use during the evolution, the impact of the plants, we know that they influence the cycle of the water and the carbon. I'm pretty convinced that it will be very interesting to have an idea about how the conductance can influence at the level of a plant in the small area, and how it can improve maybe the model for the water and carbon flux. And I would like to thank you, and I went a little bit fast.
Thank you very much, Jonathan and Nathalie. So we have time for one or maybe two short questions, and then we will have a break.
I have a question.
Ben?
I have a question for Jonathan. Can you hear me?
Yes.
So I think if I understood correctly, in some of the data you were showing us, we saw that the acclimation response was saturating in plants. I think that's what you were really saying.
Yeah.
So,
yeah.
So that's for acclimation, but then there's adaptation too, like genetic adaptation. Do you see any signs for that too in data?
Well, at least for this 5,000-year-old tree, we can probably just see it as a very, very unusual acclimation experiment, because there is no genetic change probably, but there is also epigenetics, and that's basically, I think it's an area where it's really unexplored. But for other trees, we see that basically that the response saturates. And so with increasing VPD, it's really changing a lot of things. Yeah, and you see it in the ecosystems. It's a temperate rainforest, and at least in this temperate rainforest, you see that the atmosphere is drying, and that is producing a lot of changes. And clearly, this is affecting these very ancient trees, and to that scale. So it's a strong magnitude. This tree has been responding quickly, but then the magnitude of this recent change basically is flattening the curve. And the same probably we can see with the isotopes, stable isotopes, to see a bit we are doing the analysis now for these very old trees where basically they are not growing height, so the ontogenetic change is basically frozen at this time scale, and so we can really see the adaptation of these trees or acclimation of these trees to these recent changes.